Ribonucleic acid (RNA) is a polymeric molecule essential in various biological roles. It is present in all living cells except few viruses. Its important role is to act as a messenger carrying instructions from DNA for controlling the synthesis of proteins. Like that of DNA, RNA is also assembled as a chain of nucleotides. But unlike DNA, it is found as a single strand folded structure rather than a paired double strand. Structure of RNA. The structure of RNA was described by R.W. Hawley in 1965. It is a single-stranded helix with 5' to 3' end. The major component of RNA includes pentose sugar, nitrogen base, and phosphate group. Pentose sugar consists of ribose sugar with 5 carbon atoms, 10 hydrogen, and 5 oxygen. The chemical formula is written as C5H10O5. The presence of a chemically reactive hydroxyl group attached to the second carbon atom in ribose sugar makes RNA prone to hydrolysis. This chemical liability of RNA is thought to be one reason why DNA evolved to be the preferred carrier of genetic information in most organisms. Nitrogenous bases in RNA are of two types, namely purines and pyrimidines. Adenine and guanine are purines, while cytosine and uracil are the pyrimidines. Phosphate group is involved in the formation of phosphodiester bond which links the adjacent ribonucleotides. Ribonucleosides. It is an organic molecule derived from the condensation of a nitrogenous base and first carbon of ribose sugar by glycosidic linkage. The purine are linked to sugar at 9th nitrogen atom, while the pyrimidines are at first nitrogen atom. Adenosine, guanosine, cytidine, and uridine comprises the 4 nucleosides of RNA molecule. Ribonucleotides. Ribonucleotides are the building blocks of RNA, composed of 3 parts, namely a ribose sugar, a nitrogenous base, and a phosphate group. The nitrogenous base is attached to the first carbon atom of the sugar, and the phosphate group is bounded to the 5th carbon atom of the sugar. During RNA synthesis, the phosphate group of a new ribonucleotide is attached to the 3rd carbon atom of the existing nucleotide by the enzyme RNA polymerase, thereby forming a polynucleotide of RNA. Types of RNA. RNA occurs in different forms within the living organisms and serves many different roles. In both prokaryotes and eukaryotes, there are 3 types of RNA like mRNA, tRNA, and rRNA. mRNA. Messenger RNA accounts for about 5% of total RNA in the cell. It carries the genetic code copied from the DNA during transcription in the form of a triplet nucleotides called codons. As a part of post-transcriptional processing in eukaryotes, the 5' end of mRNA is capped with 7-methyl-guanosine triphosphate nucleotide. It helps in mRNA recognition during translation or protein synthesis. In prokaryotes, 5' cap is absent. After the 5' cap, there is a section of mRNA that are not translated. This section is called 5' UTR, stands for untranslated regions. 5' UTR provides stability to mRNA, localization of mRNA, and increases translational efficiency. Following the 5' UTR, there will be an initiator codon. In prokaryotes, AUG or GUG can act as initiators, while in eukaryotes, AUG is only the initiator codon. It directs the initiation of protein translation by stimulating the binding of initiator tRNA. Coding region follows the initiator codon. Coding region are composed of triplet codons which are decoded and translated into protein by the ribosomes. In addition to protein coding, portion of coding regions may serve as regulatory sequences in the pre-mRNA. A nucleotide triplet in the mRNA that signals a termination of translation process is called stop codon or terminator codon. It is located at the end of the coding region. There are 3 codons like UAA, UGA and UAG, which serves as stop codons. The section of mRNA that immediately follows the terminator codon is called 3' UTR. The 3' UTR often contains regulatory regions that influence gene expression. The 3' end of the mRNA has a poly-A tail which prevents enzymatic degradation of mRNA. It also promotes export of mRNA from the nucleus into the cytoplasm for translation. mRNA transcribes the genetic code from DNA into a form that can be read and used to make proteins. mRNA carries genetic information from the nucleus to the cytoplasm of a cell. There are 2 types of mRNA found in the living organisms. Monocistronic mRNA is a mRNA molecule that encodes only one protein at a time. All eukaryotic mRNA are monocistronic. It is transcribed from a single gene and has one initiation and terminator codon. It consists of coding exons and non-coding introns. Hence, it must undergo post-transcriptional modification. Polycistronic mRNA is a mRNA molecule that encodes more than one protein at a time. All prokaryotic mRNA and chloroplast mRNA are polycistronic. It is transcribed from several genes. It consists of a leader sequence which precedes the first gene. The gene is followed by an intercistronic region and then another gene. A trailer sequence follows the last gene in the mRNA. It will not undergo post-transcriptional modifications. tRNA. It accounts for about 15% of total RNA in the cell. It is an adapter molecule that serves as the physical link between the mRNA and the amino acid sequences of proteins. It carries a specific amino acid to the protein synthetic machinery of the cell, that is, ribosomes. The structure of tRNA is explained by clover leaf model. tRNA molecule has a distinctive folded structure with three hairpin loops that forms the shape of a three-leafed clover. One of these hairpin loops contain a sequence called anticodons which can recognize and decode an mRNA codons. Because of the internal base pairing, the tRNA consists of hydrogen bonded stem and associated loops which often contain nucleotides with modified bases like pseudouridine, dihydroxyuridine, inosine, and hypoxanthine. The tRNA is made up of 73 to 93 ribonucleotides. It has 4 arms, namely amino acid acceptor arm, T-Cy arm, anticodon arm, and DHU arm. The anticodon arm and acceptor arms are oriented in opposite direction. The acceptor arm carries the amino acid, hence the name. The 3' end has 3 unpaired base sequences, CAA, and 7 paired bases. The 3' hydroxyl group forms an ester bond with the carboxylic group of an amino acid. The T-Cy arm contain a pseudouridine, hence the name. It is involved in binding of tRNA to ribosomes. It has 5 base pairs and 7 unpaired nitrogen bases in the loop. Anticodon arm consists of 5 paired bases and 7 unpaired bases in the loop. Among these 7 unpaired bases, the middle 3 nucleotides act as anticodons which recognizes the triplet codon present in mRNA. The specificity of tRNA resides in the anticodon site. The specificity is in recognition the mRNA codon as well as in accepting the specific amino acid coded by that codon. DHU arm has 3 to 4 paired bases and 10 unpaired bases in the loop. It contains a dihydrouridine area, hence the name. It serves as the recognition site for the enzyme which adds the amino acid during translation. About 75% of tRNA molecule possess an extra short arm of about 3 to 5 base pairs. The number of base pairs in this arm may vary, hence it is called variable arm. rRNA. It accounts for about 80% of total RNA in the cell. It is involved in the synthesis of ribosomes. Ribosomes provide necessary infrastructure for the mRNA, tRNA, and amino acids to interact with each other for the translation process. It consists of single strand of nucleotides. At some region, the strands are folded. In some regions, the single strand is twisted upon itself to form a double helix. Base pairing within the sequence forms stem loop configuration. Because of this configuration, rRNA can form tight and specific interactions with the ribosomal proteins to form ribosomal subunits. So to conclude, different forms of RNA primarily carry out biochemical reactions similar to enzymes. However, some of them also have complex regulatory functions in the cells. Owing to their involvement in many regulatory processes, to their abundance, and to their diverse functions, RNA plays important role in cellular processes.